Alright everybody, so I wanted to spend some time talking about some of the food web metrics that I use in my own personal research, and these are the metrics that were used in that Playa paper that we read in the beginning of class. And so we can explain them a little bit better now. So this assumes that you've made your food web based on and put the species and figured out you put the species on the carbon nitrogen biplot, and you so you know the carbon signature, the nitrogen signature, and you've put them all up. So you don't necessarily need to draw specific lines, right? We don't need to specifically write that this shark is eating both of these species. But the metrics I want to talk about you can use in a community-wide way to explain some things about the community. So the nitrogen range, you can get a proxy for food chain length based on what is the lowest nitrogen signature versus the highest nitrogen signature. So if you had a food chain that looked from something like this in one food web, and another one let's say you add a third predator here. Oops, that's the worst looking fish ever. But yeah, okay, let's just pretend that's a fish. Oof, that's tough. Anyway, the nitrogen range we would expect to be higher then. So we can get an idea of total food chain length or maximum trophic position based on the range in that nitrogen value. Another thing we could look at is total trophic strategies. Okay, so if you have a food web that has this many species, and what you can do is measure the area of this polygon here. Okay, let's say you get that area then, and you compare it to another food web that has another shark species, okay? And what this shark species, based on it being over here, we know it's going to be eating that fish and that fish alone. So this shark here has a unique trophic strategy, okay? So when we would look at that polygon, it would be bigger. So the area of, the whole that encompasses the entirety of the food web is bigger, so we know that there's more total trophic strategies in a food web that has a bigger area of this weird looking polygon. Now we can also look at the redundancy. Okay, so let's say we have a species that looks at this orange species, right? What is it going to be eating, essentially? What it's going to be eating, or this blue species is going to be eating just this. And, this orange species is going to be eating mostly of these C3 plants, but then, a little bit. So if we, make this arrow much thicker, so this orange fish here is going to be eating mostly C3 plants and a little bit of C4 plants. That's why it's much more to the right, right? So what we can do is look at the nearest neighbor distance. So what that is saying is, how close in carbon nitrogen biplot space is the blue species versus this orange species, or its nearest neighbor, okay? And if we find that there's a bunch of, let's now change this to just a dot, right? Where you just have two dots here. And if there's a bunch of dots of a bunch of different species here, a bunch of dots of a bunch of different species up here, a bunch of dots of a bunch of species up here, what that means is essentially, all the different species in this little cloud here are essentially trophically redundant. They're all essentially just eating, this species over here, or something from this group of dots over here. Whereas if you were to just have, a food web that looked, just like that, with only one species in each position, the nearest neighbor distance would actually be pretty far, right? So everything is super far away from each other. So the redundancy of the food web would be expected to be smaller in this food web compared to these where you have one species that's very close in carbon nitrogen biplot space. Then you can also look at the variation in these different, like the nearest neighbors, to figure out how the niches are organized and the variability in this of how those niches are organized, which tells you something about how regular and how far apart these niches are. So all of these different metrics we can use based off just of this carbon nitrogen biplot space, which is a really interesting way to look at food webs and explain patterns that we see that is very different from the very highly labor intensive following around figuring out exactly what each individual fish species is eating. All right, with that, I'll see you later.